Nobody told Valter Longo he was going to accidentally discover something that would challenge the way oncologists think about cancer treatment. He was not even studying cancer. He was studying yeast. It was the early 1990s, and Longo, then a PhD student at UCLA, was trying to understand why yeast cells die. Specifically, he was interested in what made some yeast cells live dramatically longer than others under conditions of starvation. When he deprived yeast of nutrients, something unexpected happened. The old cells died, but the younger, healthier cells did something remarkable. They did not die. They went dormant. They stopped dividing, stopped consuming, stopped doing almost everything, and in doing so, they became extraordinarily resistant to stress. You could expose them to levels of oxidative damage that would kill an actively metabolizing cell, and these dormant, starving cells would survive. He filed that observation away and kept working. 20 years later, it became the foundation of one of the most provocative ideas in modern cancer biology. The idea is this: cancer cells cannot do what healthy cells do when food disappears. Healthy cells, when deprived of glucose and nutrients, have the metabolic flexibility to switch fuels. They downregulate their growth machinery, upregulate their stress resistance programs, and enter a kind of protected waiting state. They become, in Longo's language, like the crew of a ship battening down the hatches before a storm. The storm passes, they emerge and resume normal function. The cell survives. Cancer cells cannot do this. Cancer cells are, by their nature, metabolically rigid. They are locked into a growth program that cannot be easily switched off. They depend overwhelmingly on glucose, not just as a preferred fuel, but as a near-exclusive one in many tumor types. This dependency is so pronounced that it is exploited as a diagnostic tool. PET scans, which light up cancer cells in the body, work because they inject radioactive glucose and watch where it gets consumed fastest. The cancer cells are consuming glucose at rates that dwarf their surrounding healthy tissue. They are, metabolically speaking, addicted, and an addict is not in a good position when the supply disappears. This is the biological premise behind what has become one of the most studied areas in nutritional oncology: the relationship between fasting, metabolic state, and cancer cell survival. The research is not fringe. It is not wellness speculation. It is coming from major cancer research centers: the University of Southern California, MIT, the Salk Institute, the Buck Institute for Research on Aging, and it is producing findings that are progressively harder to ignore. But before any of that, I want to tell you about a woman named Alessandra. Alessandra was 57 when she was diagnosed with early-stage breast cancer. She was not someone who had ever thought much about the biology of her own metabolism. She was a restaurant owner in northern Italy, someone who had spent her life thinking about food from the standpoint of pleasure and tradition, not biochemistry. When the diagnosis came, her oncologist in Milan outlined the treatment plan: surgery, followed by chemotherapy, followed by hormone therapy. Standard of care. Excellent prognosis at her stage. She went through it. She is today 11 years out from that diagnosis, cancer-free. But the part of her story that has stayed with me is not the treatment. It is what she did during it that nobody asked her to do and nobody told her not to do. During her chemotherapy cycles, Alessandra instinctively, because she felt nauseated and had no appetite, stopped eating for approximately 24 hours before each infusion and for 24 hours after. She just did not eat. She drank water and black coffee and waited for the nausea to pass. She mentioned this to her oncologist almost as an aside. Her oncologist mentioned it to a researcher colleague. That researcher colleague connected it to a study that had just been published, a study by Longo's group at USC showing that fasting before chemotherapy in mice dramatically reduced the side effects of the drugs while simultaneously making the drugs more effective against the tumor cells. The fasting had made the healthy cells resistant. The fasting had left the cancer cells exposed. Alessandra had done, accidentally and instinctively, what researchers were beginning to demonstrate mechanistically in laboratories. Now I need to be careful here, genuinely careful, not in the performative disclaimer way that gets inserted into health videos to cover liability and then immediately forgotten. Careful in the sense that what I am going to describe next is real, evidence-based biology that has direct relevance to how you live your daily life, but that exists in a context where cancer is serious enough that the language around it demands precision. This is not a video claiming that fasting cures cancer. It does not. No dietary intervention cures cancer. The treatment of established cancer requires medical oncology, surgery, radiation, chemotherapy, immunotherapy, targeted therapies, and nothing in this video supersedes that. What this video is about is the specific biology of the fasting state as it relates to cancer cell metabolism, and what that biology means for a preventive and adjunct practice that is available to every person watching, costs nothing, requires no product, and has a body of evidence behind it that is significant enough to have produced clinical trials at major cancer centers worldwide. If you have an active cancer diagnosis, please discuss any dietary changes with your oncology team. Not because the changes are dangerous, but because the timing, particularly relative to treatment, matters and your oncologist should be involved. If you are on medication for any condition, particularly diabetes medication, since the practice involves restricting food intake, speak with your physician before making significant changes to your eating window. With that established with full sincerity rather than legal formality, here is the biology. The state your body enters in the hours after your last meal is not a neutral state. It is an actively different metabolic condition with specific measurable effects on cell behavior, hormone levels, immune function, and this is the part the research has been progressively illuminating, on the relative survival conditions for normal cells versus cancer cells. In the first 8 to 10 hours after eating, your body runs primarily on glucose from glycogen stores in the liver and muscles. Blood glucose is moderately elevated, insulin is present, cells throughout the body are in growth and replication mode. This is the fed state. It is the state that most people in the modern world spend the majority of their waking hours in, and increasingly their sleeping hours as well, because late-night eating pushes the fed state deep into the night. Around the 10 to 12-hour mark of fasting, the point at which liver glycogen begins to deplete, something shifts. Insulin levels fall significantly. A hormone called glucagon rises, signaling the liver to begin producing glucose from non-carbohydrate sources. The body begins mobilizing fatty acids from adipose tissue. These fatty acids travel to the liver and are converted into ketone bodies, beta-hydroxybutyrate, acetoacetate, which begin to replace glucose as fuel for the brain, the heart, and other tissues. This metabolic shift is called the transition to ketosis. In mild early-stage fasting and the kind produced by a 12 to 16-hour overnight fast, it is partial and gentle. Blood ketones rise modestly. Insulin falls but not to extreme levels. The body is beginning to draw on fat stores rather than dietary glucose, but it has not made the complete transition of a multi-day fast. And in this transitional state, the metabolic gap between healthy cells and cancer cells begins to open. Cancer cells overwhelmingly cannot efficiently use ketone bodies as fuel. This is called metabolic inflexibility, and it is an intrinsic feature of the oncological transformation. The same genetic dysregulation that makes cancer cells grow uncontrollably also compromises their ability to switch metabolic fuels. A healthy heart cell in early ketosis shifts gracefully to burning ketones, actually preferring them in many circumstances because ketones produce more ATP per unit of oxygen than glucose does. A cancer cell in early ketosis is still reaching for glucose, finding less of it, and under a degree of energetic stress that healthy cells are not experiencing. This is not metaphorical. It is measurable in cell cultures, in animal models, and increasingly in human data. And the implications are significant for what happens in the hours before you eat your first meal of the day. If you eat dinner at 7 p.m. and breakfast at 7 a.m., you have spent 12 hours in a progressively deepening fasting state. The last few hours of that window, roughly 4 a.m. to 7 a.m., are spent in the deepest metabolic state of the cycle. Insulin is at its lowest point of the 24 hours. Glucagon is elevated. Growth hormone released in pulses during deep sleep is near its daily peak. Ketone production is at its highest of the cycle. And the cellular programs associated with this state, autophagy, the cellular recycling process, reduced mTOR activity, which slows cellular growth machinery, elevated AMPK signaling, which activates stress resistance pathways, are all running at maximum expression. In those final hours before breakfast, healthy cells are in a state of maximum stress resistance and minimum growth mode activity. They are the ship with the hatches battened. And if cancer cells are present, whether established tumors or the much more common pre-malignant cells that exist in virtually every adult body without ever declaring themselves as cancer, Those cells are in a state of relative energetic deprivation that their metabolic rigidity does not allow them to adapt to. The research on what happens to cancer cells during this fasting period is specific enough to go through mechanism by mechanism. First, IGF-1 suppression. Insulin-like growth factor 1 is a hormone that promotes cell growth and division throughout the body. It is an essential molecule. It drives the growth of children, the repair of muscle, the renewal of tissue. But it is also, in excess, a potent promoter of cancer cell proliferation. Cancer cells express high levels of IGF-1 receptors. When IGF-1 is present and binding to those receptors, it activates PI3K/AKT/mTOR signaling, a cascade that drives cell division, suppresses apoptosis, and promotes tumor growth. During fasting, IGF-1 falls substantially. Multiple studies have demonstrated that even a 12 to 16-hour overnight fast produces measurable reductions in circulating IGF-1 compared to a fed state with evening eating. And the relationship between IGF-1 levels and cancer risk is well established. Elevated IGF-1 is an independent risk factor for breast, prostate, colorectal, and lung cancer. When you extend your overnight fast to 12 to 14 hours, you are spending more of each 24-hour period with IGF-1 suppressed. You are reducing the cumulative exposure of every cell in your body, including any pre-malignant cells, to one of the most potent growth signals available to them. Second, autophagy activation. Autophagy is the process by which cells identify and dismantle their damaged, dysfunctional, or unnecessary components. It is a quality control system of extraordinary sophistication. The cell essentially turns its digestive machinery on its own interior, consuming misfolded proteins, damaged organelles, and cellular debris that would otherwise accumulate and compromise function. Autophagy is strongly activated by fasting, through AMPK signaling and through the suppression of mTOR, which is autophagy's primary brake. After approximately 12 to 16 hours of fasting, autophagy rates in most tissues are measurably elevated compared to the fed state. The relationship between autophagy and cancer is complex. The same process that clears pre-malignant cells can, in certain established tumor contexts, help tumor cells survive stress. But in the context of primary prevention, in the context of clearing the pre-malignant and genomically unstable cells that exist in tissue before they have the opportunity to establish as tumors, autophagy is overwhelmingly a cancer-suppressive process. It is removing the cellular garbage from which malignancy might develop. Yoshinori Ohsumi won the 2016 Nobel Prize in Physiology or Medicine for his work on autophagy mechanisms. The Swedish Academy described autophagy as fundamental to understanding cancer, among other conditions. This is not peripheral science. The cellular recycling that your body performs during the extended overnight fast has attracted the attention of the most distinguished scientific body in the world. Third, immune system recalibration. This mechanism is the one that most surprised researchers when it first emerged, and it remains one of the most compelling findings in the fasting literature. A 2014 study from Longo's group, published in Cell Stem Cell, demonstrated something that Longo himself described as almost unbelievable at first. Cycles of prolonged fasting in mice caused a significant reduction in circulating white blood cells, followed, upon refeeding, by a robust regeneration of the entire immune cell population from hematopoietic stem cells in the bone marrow. The fasting essentially signaled the body to clear out old damaged immune cells and replace them with new ones. Extended fasting of the kind used in that study, multiple days, is not what most people are considering or should consider without medical supervision. But the partial immune remodeling effect of regular, repeated, shorter fasting periods is now an active area of research. The specific cancer-relevant finding is this: immune surveillance, the process by which natural killer cells and cytotoxic T cells patrol the body and identify and destroy pre-malignant and malignant cells, appears to be enhanced in the post-fast, early refeeding state. The immune cells that emerge from the bone marrow after fasting-driven regeneration are younger, more responsive, and more effective at recognizing abnormal cells. The immune system that exists after a night of extended fasting is not the same immune system that existed the night before. It has been, to a degree, refreshed. Fourth, reduction in circulating glucose and the Warburg effect deprivation. Otto Warburg, a German physiologist who won the Nobel Prize in 1931, observed that cancer cells preferentially ferment glucose into lactate even in the presence of oxygen, a metabolically inefficient process called aerobic glycolysis, now known as the Warburg effect. Cancer cells are consuming glucose at rates 10 to 70 times higher than the surrounding normal tissue. They are metabolically voracious in a way that normal cells are not. During the extended overnight fast, blood glucose falls. Not to dangerous hypoglycemic levels, the liver's gluconeogenic activity maintains glucose above the threshold of physiological concern in a healthy individual. But it falls substantially from postprandial peaks, and it falls below the threshold at which cancer cells' glucose-dependent metabolism operates comfortably. The overnight period of reduced glucose availability is a period of relative deprivation for cells that depend on glucose as their primary fuel and cannot efficiently substitute alternatives. For normal cells, this is not deprivation. This is the natural state that human metabolism evolved to operate in, the feast-fast cycle that our species spent most of its evolutionary history navigating. Our cells are designed for this pattern. Cancer cells, with their rigidly programmed metabolic inflexibility, are not. Now, I want to be honest about where the research stands and where it has limits, because intellectual honesty here matters as much as the findings themselves. The clearest and most compelling evidence is at the level of cell biology and animal models. The mechanisms are worked out in extraordinary detail. The effects in mouse models are dramatic and reproducible. The human data, while increasingly substantive, is less complete, because studying fasting's effects on cancer outcomes in humans requires the kind of long-term controlled studies that are expensive, difficult to run, and take decades to produce results. What the human data currently shows, epidemiological studies consistently find that people who eat within a shorter daily window have lower rates of several cancer types, including breast cancer, where a women's healthy eating and living study found that women who fasted fewer than 13 hours per night had a 36% higher risk of breast cancer recurrence compared to those fasting 13 hours or more. That is not a trivial association. A 36% difference in recurrence risk from eating within a time window is a finding that should be on every oncologist's desk. Additional human data comes from fasting mimicking diet studies, where participants consume very low-calorie, specific macronutrient ratio diets for 5 days per month, designed to produce the metabolic state of fasting without complete food restriction. Trials at the USC Norris Cancer Center have demonstrated that the fasting mimicking diet alongside chemotherapy produces better outcomes in several cancer types than chemotherapy alone, with reduced side effects in the patients completing the fasting arm. These are small trials and larger ones are ongoing, but the direction is consistent and the mechanism is biologically coherent. The daily 12 to 16-hour overnight fast that I am describing is not the multi-day fasting used in some of these therapeutic contexts. It is the minimum meaningful dose of the fasting state, the dose that produces measurable IGF-1 reduction, measurable autophagy activation, measurable immune recalibration, and meaningful glucose reduction in the overnight window without requiring anything that a healthy adult would consider dramatic or difficult. Here is what this actually looks like in practice, because the gap between the biology and the daily life decision is where most interventions fail. If you finish dinner at 7 p.m., a 12-hour fast means eating breakfast at 7 a.m. This is not unusual. This is what breakfast means, the break of the fast. The problem is that most people are eating later and later in the evening, pushing their last meal to 9, 10, even 11 p.m., and then eating breakfast at the same time they always have. The overnight fast shrinks to 8 or 9 hours. The metabolic transition to the ketogenic, autophagy-active, IGF-1 suppressed state barely begins before it is interrupted by morning eating. The intervention is therefore not adding a fast. It is protecting the fast that already exists by moving the last meal of the day earlier rather than the first meal later. Eating dinner at 6:30 or 7 p.m. instead of 8 or 9 p.m. is not a dramatic dietary overhaul. It does not require different food, different portion sizes, or a different relationship with what you eat. It requires only that the eating stops earlier. This one change, an earlier dinner consistently applied, extends the overnight fast from 8 or 9 hours to 12 or 13 hours. And across that additional 3 to 4 hours of fasting, the biology described above is running. IGF-1 is lower. Autophagy is higher. Blood glucose is lower. Ketone production has begun. The metabolic conditions that cancer cells find inhospitable and that healthy cells find restorative are persisting for longer. The second component, which amplifies the overnight fast's metabolic effect without extending it, is what you do before breakfast. Not what you eat, but what you do physically before the fast is broken. Light physical activity in the fasted state, a 10 to 20-minute walk before the first meal of the day, produces several metabolic effects that are additive to the overnight fasting state. AMPK, the cellular energy sensor that is already elevated during fasting, is further activated by exercise. Ketone production, already underway in the late fasting period, is amplified by exercise-induced fatty acid mobilization. And crucially, the GLUT4-mediated glucose uptake in skeletal muscle during fasted exercise draws blood glucose down further, not dangerously, but meaningfully, maintaining the glucose-deprived environment that the cancer-suppressive effects depend on for additional minutes before the first meal arrives. The fasted morning walk is not primarily a weight loss intervention, though it does support fat mobilization. It is a metabolic extension of the overnight fast that costs nothing, takes 15 minutes, and compounds the biological conditions created by the extended overnight window. Breaking the fast correctly matters too, though less than the fast itself. The standard Western breakfast, cereal, toast, fruit juice, pastry, is a rapid glucose delivery mechanism that abruptly terminates every metabolic benefit of the overnight fast within minutes of consumption. Blood glucose spikes, insulin surges, IGF-1 begins to rise. Autophagy brakes are applied through mTOR reactivation. The fasting state collapses. Breaking the fast with protein and fat, eggs, full-fat yogurt, avocado, nuts, smoked fish, produces a much slower glucose entry into the bloodstream, a lower insulin response, and a more gradual transition from the fasting metabolic state. The benefits of the overnight fast are not abruptly terminated. They are tapered, allowing the early morning window of maximum autophagy and minimum IGF-1 to persist for additional hours. This is not about eating perfectly or eliminating carbohydrates from your life. It is about understanding that the transition out of the fasting state, the moment you break your overnight fast, is a metabolically significant inflection point, and that the composition of what you eat at that inflection point determines how abruptly the cancer-suppressive metabolic environment of the night is replaced by the growth-promoting environment of the fed state. Let me tell you about what Alessandra does now, 11 years after her diagnosis. She eats dinner before 7 p.m. She walks for 20 minutes every morning before she eats anything. She breaks her fast with eggs, olive oil, and vegetables from her restaurant's kitchen, not because she has been following a protocol someone gave her, but because the research she read in the years after her diagnosis confirmed the biology behind the instinct she had followed during chemotherapy. She understood, after reading Longo's work and the subsequent literature, why her accidental 24-hour peri-chemotherapy fasts had aligned with the direction the science was pointing. She does not believe she is cancer-proof. She goes to her oncology follow-ups, she gets her imaging done, she takes her hormone therapy. She is not using lifestyle as a substitute for medicine. She is using it as what it actually is, a specific, evidence-supported modulation of the metabolic environment in which her cells exist, informed by the best science available. She also thinks of her overnight fast, the 12 to 13 hours between her 7 p.m. dinner and her 7:30 a.m. walk, followed by breakfast, as something she does for herself rather than against cancer. She does not frame it as deprivation. She frames it as a nightly practice of cellular housekeeping that she happens to do while asleep. That reframing matters. Not psychologically, though it helps there too, biologically. Because sustained lifestyle practices require a motivation that endures beyond the initial information surge of watching a video. The people who maintain extended overnight fasts for years are not primarily the people who are afraid of cancer. They are the people who have come to understand that the 12 hours after their last meal are 12 hours in which their bodies are doing extraordinary, complex, self-protective work, and who have decided to protect those hours rather than interrupt them. Your body knows what to do in the dark, in the quiet, in the absence of food. It is known since long before anyone gave it a name. It cleans, it repairs, It recalibrates. It examines and removes the cellular mistakes that accumulate over a lifetime of living. The fast that happens between dinner and breakfast is not empty time. It is one of the most biologically productive periods in your 24-hour cycle. Protect it. Extend it where you can. Break it thoughtfully. Walk before you do. The research will keep adding detail to this picture. The clinical trials will run and report their findings over the next decade, and the picture will become more precise. But the core biology, the metabolic inflexibility of cancer cells, the autophagy activation of fasting, the IGF-1 suppression, the immune recalibration, the glucose deprivation in the overnight window, this is not provisional. This is mechanism. This is the established biology of what your body does when the kitchen closes and the night begins. Valter Longo studied starving yeast in a UCLA laboratory in the early 1990s and noticed that the healthy cells survived and the compromised ones did not. Your overnight fast is the same experiment. Running every night, inside you. Let it run.